right, so good afternoon, everyone. A big thank you to our three speakers who took some time out today to come here, visit John Abbott College, and talk a little bit about our research, I should say. And a big thank you to all of you for attending and being such a captive and wonderful audience. So, I guess the way we're going to work this Q&A panel is, I'll ask you guys first. You're the audience. I want to hear from you guys. Like, do you have particular questions about the research? But part of this is also about learning about the careers, maybe the career directions that these three young scientists are sort of carving out for themselves. So if anyone has any questions, anything at all, anybody at all,
So I was wondering, does animal welfare physically impact the quality and the nutritive content of the food that we eat?
That's a question. So
Yes, 100% yes. There actually was a study that recently came out, it's yet to be validated, but it's called Happy Cows Equals Happy Milk. So we have found both with physical and mental welfare that having animals who are more comfortable, who are less stressed, who are in environments where they have control and agency over what they do does impact the product. Even when we're having animals in a slaughter facility, for example, like pigs, we want them to be as calm and relaxed as possible because if they're stressed, that actually makes the meat taste not as good. So yes, welfare is very important not only for the animals themselves, but also in the products that we purchase. It does make a difference. Thank you for your question.
I have to give it to Felix, guys. He sent me a check.
Okay, so I have two questions. One to the presentation on animal welfare. I wanted to find out, since we're talking about sustainability here, allowing the animal some kind of mobility, what effect does it going to have on the land use or land occupation? And then my second question is to the presentation on oats. I think I saw a part that talked about using mutation to achieve the gene editing. I want to find out, is that mutation controlled? And if no, does it have any adverse possible impact on the results that we're going to get in the end? Thank you.
All right, thank you for your question. So yes, some producers, some farmers don't necessarily have the land to be able to give their animals outdoor access, which is why before in our code it wasn't recommended or regulated. But now, as we're giving them time, we're hoping that they will be able to provide accommodations, but that's why it's still a recommendation to allow them outdoors. The actual regulation, which says they have to be able to have some movement, doesn't necessarily mean movement outside, but they can still have indoor movement, so unchaining them and allowing them to still move around the barn. But in terms of land use, that's something that is potentially a problem because farmers don't necessarily have the land to be able to provide them outdoor access, which is why it's just a recommendation to start, but we're hoping as time goes on that they'll be able to provide that accommodation, but it's something that still needs work.
Thanks, Jasmine. Mehtab.
Thank you for your question. That's a valid concern. So you don't want to alter oats flowering but mess up some other gene that is having some other effect on the oats. Yeah, so when we design our guide RNA for the experiment, we do off-target analysis that makes sure that that same target is not found in the oat genome somewhere else, so that the guide can bind to other gene and then also knock out that. So we make sure there is no off-target effects, and also before the product is released, there is a whole genome sequencing people do to see if the product that's made just has the change on the gene that you are targeting but not somewhere else. So all the oat genome is intact except the gene that you are working on.
Yeah. Mehtab, I'll ask a question before I go to the middle of the room. I know there's some people who have their hands up, but on a related note to that question, are you worried about, and again, pardon a naive question, but in editing the gene sequence, are there concerns about maybe the organism's sort of ability to adapt to other stresses that might present themselves in the future? If we kind of edit and then selectively choose to grow that particular oat, if a new stress comes in, might it be less responsive to that new stress?
That's an interesting question. It can be yes, it can be no. It depends if that gene is responsible for, like there are some genes that are pleiotropic, so they can have more functions than one. So if this gene is not only targeting the flowering time, but also responsible for drought tolerance, or but it depends. If the gene is overexpressed, then it's drought tolerant, or it's underexpressed, that's drought tolerant. So is it promoting the tolerance, or it's not promoting it? Yeah, but this gene is related to just yield and flowering time, and that's also we're trying to understand using gene editing. So what's the exact role of this gene? How, up to what extent does it alter the flowering time? Yeah, but I mean, we have to have those plants and control them in some stress to know that it's behaving differently.
Perfect. That was a question? I'm sorry. If you feel like you got the
I'm
pipes.
just wondering, when it comes to the cow frustration, you had plotted, let's say, cow frustration over the amount of weeks, but what determines the cow frustration? Was it, because I saw it with like percentages, is it just subjective, or were there certain things in the cows you're looking for?
Thank you, fantastic question. Yes, it's not just looking and saying, oh, well, they look frustrated. We actually have a very robust way of going about it. So we record the animals while they're in the stall, and then we have what's called an ethogram, which is basically just a table which tells us the different behaviors, and then I have to come up with the definitions of what those behaviors look like. It has to be very, very specific. You can't just say, oh, defecating is cow poops. You have to actually say, okay, when the cow lifts their tail and fecal matter comes out, and et cetera, et cetera. So what we do is first look at the behaviors that they do, and then we also look at the activity level, because when you're highly frustrated, you're going to do more activity in response. So we take both the behaviors, for example, if we see them foot stomping, scraping their foot on the ground, pulling at the chain to try to get towards the door to go out, those kind of behaviors, as well as if they're doing a lot of activity, both of those together will tell us that the animal is frustrated. So we have to take both indications. If they're just doing one or the other, maybe it's frustration, but it's better when we have all of that to tell us, okay, most likely this animal is not happy with the situation. So not necessarily that it's frustration, it could also be some other highly negative emotion, but most likely it's frustration because we saw in other parts of my project, we looked at the motivation to go outside, and they're very motivated. They will run, they will jump, they will push to get outside. They will fight other cows to be the first ones to go out. They will push past humans to go out, so we know that they're very strongly motivated for the outdoors. And then we saw, once they were kept inside, that they did present those kinds of behaviors. Not just the percentages, but yes, the fact that the behaviors themselves were present, how many times they did it, how often, etc. So all of that combined tells us that this is likely frustration.
Are there any? Oh, there we go. Excellent. Now, again, it doesn't have to be always a scientific thing. It's particular to science. If it's something that's more general, guys, I encourage you to ask any question, right?
I was just wondering, with the study on fish and all the samples, is it different that you're observing the effects of the chemicals on a short period of time on the young fish versus like a prolonged exposure to the chemicals in like the more adult fishes? Like, I understand that it's more efficient, but would the results be different if you're looking at it over a longer period of time, or?
It's a really awesome question. Here you go, Aylish.
Yeah, I lacked the time to really get into details during my presentation, but the reason why we're looking at genetic signatures is because we're trying to see if changes in gene expression can suggest or hint towards long-term, what we call chronic effects. So we're looking at, are there changes in genes that are related to gamete production, for example? That could suggest that there's going to be some reproductive issues. Obviously, there's certain things that could happen in adults that would not happen in like embryos or juveniles. That is always something to consider, and it does happen. I wouldn't say common, but it does happen, especially in birds, for example, that the adults are more sensitive than the young. But in general, for fish, the life stages that are at the earliest, so when they're freshly hatched or when they're juveniles, they tend to be a lot more sensitive to chemicals than their adult counterparts, because they're in the process of development. Their skin is a lot thinner. They rely on like food a lot more, or as far as like any contaminants in their food affect them a lot more. So they are a good substitute as far as doing kind of initial tests, but they're not complete substitutes for adult tests that are longer. But we're just trying to see if we do these tests really quickly in the young and we don't see anything, that probably suggests that we might not need to do anything in the adults. But if we see something in the young that could be severe, and we see that there's potential for long-term effects, then we can set those aside to be able to do those long-term effects. It's kind of more of a sparsing tool.
Oh, there we go, Ben.
You had mentioned how there were like 3 chemicals that were making their way into the waterways. If let's say you determine that they are having a big effect on the fish, are there measures that are put in place right now to stop those chemicals from being in the waterways? Or is it just like kind of just, it is what it is and we'll try and fix the fish instead?
In a lot of cases, there's no way to fix the fish. If the levels are high enough, there's kind of 2 things that can happen. Either we try to limit the amount that goes in the future, and then we hope that it'll be like dissolved out kind of thing. Like the more water that gets put in with rain and stuff will kind of dilute the whole substance, and then it will be less, it won't affect fish as much. But unfortunately for the example of the tire chemical, this was actually a huge issue in the last few years, where 6-PPD was having significant effects on fish like salmon in BC, for example. There was massive die-offs and we didn't know what was happening, and we were able to identify that as the chemical, that it was coming off of tires and being washed away into creeks that the salmon were using to travel. So in that case, there was a huge ban on the chemical immediately. There was recalls of tires that were particularly susceptible to leaching these chemicals. So in general, it really depends on what kind of chemical. Again, a lot of them are pre-approved for certain uses. Like a lot of agricultural chemicals are tested on insects, mammals, the plants that they're being sprayed on, things like that. So there is initial testing. Like there's never well, I won't say never, but there's very rarely chemicals that are just used with 0 testing. But especially when it comes to fish, they're pretty understudied when it comes to chemicals, especially for like genetic effects. So in that case, it's really important to do these kind of more in-depth tests. So in general, we hope that the effects are going to be low, or that the threshold is higher than what already exists. But in the case where that threshold is not adequate, then depending on the severity, we can make recommendations to the government or to appropriate departments, and they make the decision whether to ban the source or reduce, like do emergency reduction of use for farmers and stuff like that. So unfortunately, we don't make any decisions, but we do make recommendations based on the findings that we get.
All right, we've got 2 questions over here. This is great so far, just saying.
So I know the chemicals, obviously they affect the fish, but would there be a risk posed to people who eat the fish possibly?
Yeah, okay. Then I'll be back. Oh yeah.
It really depends on the chemical or on the contaminant. I mean, we all know the case of mercury, where there's pretty significant bioaccumulation, where if there's a lot of mercury in the water, then it does stay in the tissues long term. Some of the chemicals that I research, they are detoxified by your system. They don't tend to stay as much. But again, that's not specific to my research. So as far as them, if there's some of these chemicals where I haven't seen that research, I couldn't tell you. But in general, if there is bioaccumulation and there's found to be contaminants in the fish, that is a good reason why they would ban those chemicals. Or they would ban, let's say, fishing in certain areas and tell people not to consume those fish because it could be hazardous to their health. Like in cases where they've suggested recommendations on limits for the amount of certain large fish species to consume, like tuna, where they're saying don't have it more than a certain amount of time a week, because we can get rid of the metals over time, but if we eat it too frequently, then it does accumulate.
For the fish that you would, the endangered fish, I think it was the redstone something. How exactly are you able to keep track of them, and how do you get a sample of them since they're considered endangered? So are they pre-tagged? Do you do sampling, etc.?
That's a good question.
So in a lot of cases for for fish, it's harder to tag them. And in a lot of cases, especially if they're not like massive, massive fish, we don't tag them just because the potential for either infection or disruption of their regular activities is too high. So in the case, so that type of fish is called the copper red horse. And basically what we do is that because we only need the eggs, we don't actually keep any of the adult fish. And I don't think the government would let us if we tried. So basically what we do is that we go to a dam that's on the Richelieu River, and what we do is that we use the fish ladder that's there. So it's a way of fish to bypass the dam, and what we do is that we put a net at the bottom of that ladder with holes of a certain size. So any smaller fish can go through no problem, but big fish, such as copper red horse, they get stuck. So as soon as we feel a pull, we bring them up, we check them, and if they're the right species, we keep them. And then what they do is that they are able to inject them with naturally occurring hormones that stimulates the production of either sperm or eggs, and then they do a lot of artificial insemination. So part of those go to us for helping them because we do provide manpower. And a lot of those are reared until they're a certain size where we kind of help them bypass the most dangerous part of their life cycle where they're at the most risk of predation, and then they go back into the river. So that's been a huge way that those numbers have been able to increase, and then kind of as a quid pro quo, we get some of the eggs to be able to do a whole bunch of different kinds of research to try to get more information on them to see if there's things that we're missing that would explain why they've had such drastic declines in population numbers in the past. Because we're still not quite sure. We're trying to figure it out, and if we're able to figure it out for this type of fish, if it's chemicals, if it's anything else, then we might be able to extend that to other species in the region or neighboring lakes or rivers that are also having issues.
This is why Mehtab works with oat. It just stays put. It's so much easier. Is there another question? Oh, wow. Standing room only now. Here we go.
This is for the punctual oats. So I know the whole research was to make the flowering time earlier in the year. Would that somehow affect like the pollinators of any sort? And a second question also for you is, I know there's like a field of study where people they add like vitamins or whatever else into food. Like in places that, let's just say, are lacking this type of vitamin. Is that the same field of study that you're in, or?
All right. Big of a top.
It seems like there are more fish eaters than oatmeal in the room. A lot of questions on how fish can survive the chemicals, yeah. So for the pollinators, oat is the self-pollinating crop, so it doesn't need any pollinator for to carry the pollens and fertilize it. So it just happens in the closed flower. It doesn't open, it just pollinates and then it opens up. So there's no need of pollinators as such. And I think you're talking about biofortification, in which you add certain vitamins as a golden rice, where vitamins, I think it's A, was added so that it's rich in vitamin A as a product. So yeah, we're not working on that side. And it's not about expediting the oats flowering, because we are targeting a gene that is responsible for flowering time. So altering the gene might delay or expedite the flowering, but both of those germplasm, our goal is to create the diversity at that germplasm, and then for the areas in Canada where their snow takes place like later in the year, so we can have more like the oat crop which lasts for longer, because more it lasts on the field, the higher it's yielding. So where, like in Western Canada or somewhere in Saskatoon, Winnipeg, there's chances of getting snow earlier, where like there's a lot of snowfall. So in that cases, we will recommend the varieties that are early flowering, which would also come out of our germplasm, so that they can expedite, they can flower on time, and then they don't need any glyphosate or such chemicals for expedite flowering, which is not desirable. I hope it answers your question.
Perfect. So, well, I'm looking at the time, and I do want to ask a couple of general questions, and then if we have time, I'm totally going to throw it back, okay? Because part of, again, the symposium, it's a lot about the science, and there's some great questions are coming from the audience, but it's also to learn a little bit about like how we came into doing science and why we chose to do science. So I have a general question for the panel, and I'll start with you, Mehtab, and we'll just work our way down to Jasmine. What inspired you to pursue science as a career?
That's an interesting question. I didn't think of it until I was in my high school. So my father, he's the professor, he's a retired professor. So I've been looking how he has been working in the lab, in the fields. And I used to go to the fields with him, and I would see like why some of the plants look, they're still similar but different in their own ways. Like I showed the oat doesn't have like pink flowers. It do have a flower, but it's a biological flower, doesn't look pink. And also in the cereals, there's a lot of diversity. So I was like, why do the plants look similar but yet different? What are those genetic or molecular mechanisms that are making them look different in a way? So that really captivated me to pursue plant science as a career. But then in my undergrad I did bachelor's in agriculture. So I read all other different courses in agriculture, not just the biotechnology or molecular biology that I'm doing right now. We studied soil science, horticulture, flowering, floriculture, and then you have extension where you deliver your research to the audience, what you are doing. There are a lot of the entomology working on insects, and there is part of animal science as well. Yeah, so but this was the field that I was really captivated to, and then that's how now I know how the plants look different because of the gene expression, because of the epigenetics, and what environment they are growing in, and how some of those plants are very, tough. They can even grow within the concretes, and some of them need proper watering to grow, but at the end of the day, both of them are plants. So that genetic diversity, which was created naturally due to limitations in the genes and all other environmental factors as well. So I was really motivated to explore all of these, and unfortunately got a similar project to work on.
Well, I'm assuming that this is like a similar motivation to the reason why a lot of people are in the science department at John Abbott, but I had a pretty large sense of curiosity as a kid. I'm sure other people have been called know-it-alls, but it's just a factor of being very curious and having a relatively good memory. But yeah, I was always like reading different things. I had this set of like Disney encyclopedias that my parents bought me, and I must have read them like 10 different times. But yeah, it was just a matter of like wanting to learn more, and the fact that I was constantly hearing about, even in high school, so many different new discoveries that were happening, and it was so often in biology that we were learning so much so quickly. And then you learn more about like everything that had to be discovered to lead up to what is now being talked about. I just thought that there was such a untapped, well not untapped, but like currently being tapped potential to develop new technologies or make new discoveries, and I just really wanted to be a part of that. I wanted to find out all the things that I didn't yet know, and I thought that this was going to be the most fun way to go about it, I guess.
Thank you. Yeah, I guess I was kind of similar. I was always so curious. I always wanted to learn more. Growing up in school, my favorite subjects were science subjects, and I always enjoyed understanding how things work and not necessarily like why, but a little bit of both. And I think it kind of spurred me into, okay, well, what kind of field is one where I can always find out new things, where there's always new things to learn and to work on. And of course, that's what science is. It's always emerging. There's always new things that we don't know about that we can find out more. And that was something that I was really interested in. So it kind of helped me set the path of, okay, here's where I want to go. And it's either that or theater, which of course, if you know me, you can tell that that would have been a great career as well for me, but I think science is something that I really enjoy, and every day I'm excited to wake up and find out more. So if you're interested in science and you're interested in a career where you'll get to do something different every single day, I would definitely recommend it because it's so rewarding, it's so fun, and you'll meet some really interesting and intelligent people who will teach you and help you to grow as a person as well.
I'll let you hold on to the mic because I'm going to use my teacher voice now. Did any of you go through the SASA program like in Quebec? Yes, you did? Awesome. Can I just maybe pass it to Aylish? What changed about your perspectives of learning science when you started research versus when you were in the shoes of these students here. You know, sometimes we're guilty of, in science education, we spit a lot of facts at you, and yet when you actually do science, you don't have all the facts. Maybe if you could just give a quick comment about that.
Yeah, I think that one of the most important things they don't tell you in CEGEP is the only people that you really hear about in science are either your professors, who are a lot older than you and seem to know everything, or Nobel Prize laureates, who are a lot older than you and seem to know everything. So the average person working in science, if they're any good, will be more than happy to tell you about all the things that they don't know. And part of science is winging it. I mean, you're trying new things, you're seeing if it works. If it doesn't work, you're thinking about why it didn't work and what to change and then redoing it. So I think in CEGEP, it gives you an impression that you need to know what you're doing or exactly what you want to go in to be able to go forward in science, and that's not the case. I mean, there's a person in my lab who was in vet school and decided that he didn't really want to be a vet anymore, so he decided to start a PhD in toxicology, which is, you know, quite different than going to vet school. When I applied to McGill, I was applying in biology and physics, which is two wildly different areas of science. It's just a matter of like knowing that nobody is better than you, or nobody is, you know, more suited to go into this than you are, of just having some confidence of, I don't need to know everything, because the whole point is not going into it knowing everything, but going into it with an open mind and being willing to learn from everyone around you, and wanting to become somewhat of an expert in something. You don't need to know what that something is, but you just need to have that curiosity of wanting to learn more about topics. And you can learn what you want to do as you go through your undergrad. You really don't have to know it before you enter school. There's tons of opportunities to change, to discover new areas of science. I definitely didn't know about toxicology in CEGEP. So I think it's just like keeping an open mind, but also not comparing yourself to other people as much, especially not people who have been doing this kind of work for way longer than you've been doing it.
Okay. And then I'll just flip it to Jasmine, just sort of on a related note. Like, so the median age of a CEGEP student is probably about 18. Is it too early to get involved in research?
Definitely not. And I remember my first year of university, I had some friends who were saying, oh yeah, I went and talked to the professor and they said they had some positions open for the summer and yada yada. And I was like, ooh, you went to talk to the professor, that's scary. Like, I don't think I could do that, because they do seem like this big important person who has their own research going on, and you know, it's a little bit nerve-wracking. But there are lots of opportunities. First piece of advice, don't be afraid to talk to your professors or your teachers, to ask them, hey, do you have a position available? Do you have something I can volunteer to do? That was actually how I kind of made my way into research, is I just reached out to a professor whose class I really liked, and I said, hey, like, I'm interested in research maybe, I'm not 100% sure, but do you have something that I could help you with? And she had a grad student who was looking for a helper to watch mice behavior, which I hadn't worked with mice before, and I got to watch them basically play, which was really cool. And so I would recommend not to be afraid to ask if there's positions open, and to be proactive in looking for those positions. Sometimes your school will have a central website where they'll have positions posted, sometimes volunteer, sometimes paid. You can even go to different areas, different places. Like I went to a vet clinic and just said, hey, I'm looking to volunteer for a few hours. So you can always go find opportunities out there. You just have to be a little bit proactive about it. But you can start getting experience as early as now. The earlier the better. But I think there's no right way to find research. You just have to keep looking and trying to find what you're interested in. And even if trying something, you realize, oh, this isn't the right opportunity for me, or maybe I didn't understand or like this research as much as I thought, that's okay. At least it tells you something, and you can keep trying to find other avenues. So start now if you can, but as you're going, just keep in mind there's lots of things out there for you, and you can always find something.
I'll throw one last question at Mehtab. Mehtab, are you doing your master's or your PhD right now? PhD, okay. So just because, again, these kids are, you know, they're on their trajectory, just starting. Like, what's next?
PhD.
Yeah, I mean, like, have you thought about it? Have you thought about what the prospects are? Like, what can you do if you have this PhD in a domain where you're so specialized in books?
Yeah, that's interesting. Okay. So either I can go in academia or I can go to industry. So having a PhD is a good asset if you want to go in academia. You can apply for professor positions if you are like interested in opening up your own lab, working on the concepts or ideas that you have been fascinating you since your childhood, or maybe when you started your grad school and you didn't have that freedom to try up all those ideas, and you have freedom to try everything once you are a professor. And secondly, if you have a PhD, you have a good scope for in industries as well. You can be research lead, project lead, and then you can be directors and the leading roles of in the industries where you coordinate. So you kind of get those leading roles if you have a PhD. That's what I've heard from my friends who work in industries. So it's easier to get into industry if you have masters, because sometimes you might be overqualified if they're not looking for that high degree for that small type of work. But if you are able to get into industry even after a PhD, you have high chances of getting promoted to those lead roles where you are project lead and then you have a team working under you. And also after masters, if you go into industry, there are some of my friends who got the opportunity to do a PhD in an industry. So industry will fund your PhD, find a supervisor with a school, and then you can do your PhD while working in industry as well. So yeah, these are the two perspectives that I think of. And also, if nothing works out, I think I'm really good at photography, so I'll open up and do photography stuff. So.
There we go. I am going to give the last question. Let's end with some science, maybe.
So I was actually curious, the idea of introducing genetically modified organisms, is there a risk of it interacting with like the pre-existing ecosystem? Like say, for example, they escape a controlled environment, since with crossbreeding it can be quite unpredictable.
Waiting for this question, and this is probably the last question. I think so. Okay, so there's a difference between genetically modified organisms and gene editing. So in genetically modified organism, you introduce a gene into the genome which is not present already. So in this, we are just tweaking the gene, which has been going on in nature since evolution. So plants have been mutating themselves, and there is natural selection. And then so it's sort of like expediting evolution in a way. So you're not introducing anything in the genome which is not present at all. Like there is Bt cotton or there's Bt brinjal, where you introduce a Bt gene which provides resistance from certain insect. So it has a gene which is not from plant. That is a transgenic, that is genetically modified organism. Definitely it's a topic of discussion as well and dispute among the science community as well as the people who consume it. But in case of genetically modified, in case of gene edited, it's definitely approved by Health Canada as well. So once you are able to get out that CRISPR-Cas9 cassette from your plant, so it's transgene free now, but has the alteration in the gene that you're working in, and once it's homozygous, there is no risk of interacting, and also it is a self-pollinating crop, so it's not, it's like maize, or the pollen is not going to land up in some other plant, which is even in isolation. But anyways, even if it is, it's still homozygous and it's approved by Health Canada. So the plants which are gene edited and are transgene free, they're not regulated and they're open for consumption and growing in the environment. So there is no risk. I'm sure if you understand like the difference between GMO and gene edited, which is, yeah.
All right, so I think we've reached the end of our session. I invite any of you to still go grab a snack, a coffee, a drink. You can come talk to our wonderful speakers. Before we close out, just a few acknowledgements. I just want to thank again McGill University and specifically the Lister Family Initiative for supporting these students coming here, being able to share things here with our students here at John Abbott College. A big shout to Ingrid Shiraz and to Andrew Churchill, who helped organize all this. It's Churchill, right? I caught it right. I saw the look, I was like, uh-oh, I messed up the name. But no, I got it right. But a big thank you to both of you for reaching out year after year and coming back here to John Abbott College and giving your students an opportunity to talk to the public Again, thank you to Krim Jaffer, who as Honor Science Coordinator is always looking to bring these kinds of experiences to our students here. And just a shameless plug, May 3rd, we're back here in the same bat time, same bat channel. Maybe you don't get that reference, it's okay. But we are here again May 3rd in the library for another series by the Lister family. Oh, look at that. There we go. Thank you, sir. All the deets are there. And thank you to our three speakers. If we could just give them another round of applause, please. And thank you to all of you for coming and the wonderful questions that you asked. Have a great Friday, and we'll see you soon.
Thank you.